Welcome to the last video in the Bacterial Metabolism unit. In this video, we're going to look at the chemolithotrophic and phototrophic metabolisms and compare those to the chemoorganotroph metabolisms we've been talking about in the past videos. We'll also touch on anabolic processes, but we're not going to cover those in a lot of detail. Let's take a look. Chemolithotrophs are organisms that obtain their energy from chemical substances. However, unlike chemoorganotrophs that you are used to learning about, these organisms do not use organic molecules for their energy source. Remember, organic molecules are things like glucose and carbohydrates and lipids, things you normally associate with energy. Because these molecules are not broken down for energy, chemolithotrophs do not run pathways like glycolysis or the TCA cycle for ATP production. They do, however, have an electron transport chain. And all of the ATP that they produce to use in the cell is produced through oxidative phosphorylation from that electron transport chain. Instead of using organic electron carriers like NADH and FADH2, chemolithotrophs use inorganic chemicals as the electron source for their electron transport chain, such as iron or nitrites or hydrogen gas. Because of this, they often get the nickname of rock eaters, since they grow off the inorganic molecules just found in the environment. It's important to note that the electron transport chain functions in the same manner with the same steps. It's just the source of electrons that differs for chemolithotrophs. For example, we can take a look here. This would again be the cell membrane. Here are the proteins of the electron transport chain, along with ATP synthase. However, instead of NADH dropping the electrons off, we might have a molecule such as nitrite, which is going to drop its electrons off. When that happens, this forms nitrates as a base product. So one, we still drop our electrons off, but notice it's not NADH. Step 2, those electrons are still passed along the chain. Step 3, we're still going to pump protons out of the electron transport chain. And step 4, those electrons are still going to be passed to a terminal electron acceptor. And chemolithotrophs can either be aerobic, where they're being passed to oxygen, or they can be anaerobic, where they're being passed to something else. This proton gradient that's being produced will again diffuse through ATP synthase, powering it to produce ATP. That proton gradient is also used to power the flagella and various transport mechanisms, just like we saw in the chemoorganotrophs. When you look at the anabolic characteristics of these organisms, most of them are autotrophs, meaning that they build their own macromolecules from inorganic carbon dioxide. This means that these organisms never need to consume organic molecules at all. They don't use them for energy or to make building blocks for their own macromolecules. While the majority of chemolithotrophs are chemolithoautotrophs, it should be noted that there are some chemolithoheterotrophs that do use organic molecules to build their own molecular structures, just not to get energy from. All organisms use redox reactions in their catabolic metabolism. The main difference between a phototroph and a chemotroph is that chemotrophs use electron sources that already contain high energy electrons, which means those molecules are able to pass the electrons off to another molecule and release energy in the process. In phototrophs, however, the electron source contains low energy electrons. They're unable to be transferred. Phototrophs harvest energy from the sun to excite those electrons, transferring the kinetic energy in the photons of light to the low energy electron, making a high energy electron that is now able to participate in redox reactions to generate ATP. All phototrophs contain an electron transport chain that functions the same as the electron transport chain in chemotrophs. The only difference is that the electrons must first be excited in a photosystem before being passed to the electron transport chain. Here we see a low energy electron being dropped off in photosystem 2 by water. The photosystems are where the light-absorbing pigments are. Things such as chlorophyll are sometimes found in phototrophic bacteria like cyanobacteria, but other light-absorbing pigments also exist in bacteria that don't exist in eukaryotes. These light-absorbing pigments absorb the photons of light and then transfer that energy to the electron, causing the electron to gain energy and be ready to enter the electron transport chain. In the electron transport chain, that electron will now get transferred from protein to protein, just like in the chemotrophs. As the electron is transferred, it releases a little bit of energy that is used to pump the protons outside of the cell. That proton gradient can then pass through ATP synthase, just like we saw in chemotrophs. Also like chemotrophs, that proton gradient that has formed outside the phototrophs can be used to power things like their flagella and nutrient transport. So not all of it goes to ATP synthesis. Some phototrophs have two photosystems, such as shown here in this image. But others just have one. No matter the number of photosystems, at the end of the transport, the electrons still have to be removed from the electron transport chain, just like in chemotrophs, using a terminal electron acceptor. In this case, that terminal electron acceptor is NAD+, which picks up the electrons, forming NADPH. One thing to note is there's a difference between phototrophy and photosynthesis. Phototrophs use this light-powered electron transport chain to make ATP to power all of the bacterial cellular processes. Some of these organisms are autotrophs, also using carbon dioxide to make molecules. But some are heterotrophs and use organic molecules. This is different than photosynthesis that you may have learned in plants. Photosynthesis is actually a combination of photophosphorylation with the electron transport chain, what we've just seen here that happens in phototrophs, and biosynthesis from carbon dioxide as an autotroph. Photosynthesis is really photoautotrophs. The ATP made during photosynthesis in a plant chloroplast is only used to power the autotrophic carbon fixation that's also happening in that chloroplast. But the ATP made during photophosphorylation of a phototroph is used to power the entire bacterial cell. We have not discussed the details of the anabolic reactions in this unit, and due to time limitations, we're not going to be. The anabolic reactions start differently in autotrophs versus heterotrophs, but once the main building blocks are generated, many of the pathways are the same in these organisms. These pathways are similar to some of the chemoorganotrophic catabolic pathways we looked at already, but there's typically some unique steps. For example, to build a molecule of glucose, taking pyruvate and working up the pathway to make glucose, as opposed to taking glucose and breaking it down to pyruvate, this pathway is called gluconeogenesis. And if you notice along the way, there are many steps where we have the arrows back and forth that are the same for both glycolysis and gluconeogenesis. But there are a couple, such as here at the bottom and these two up top, that are unique to gluconeogenesis. There's a different enzyme powering this step than what was powering this step. These steps that are unique to the anabolic process allow the cell to better regulate whether glycolysis or gluconeogenesis is happening in that cell. Remember that anabolic processes must build all of the cell structures, including peptidoglycan, cell membranes, lipopolysaccharides and gram-negative cells, enzymes, nucleic acids, and all the other macromolecules of the cell. To do this, the cells first have to build a carbon skeleton, and then they're going to assimilate nitrogen or phosphorus, those other atoms, into the organic molecules, depending on what is being built. This image shows some of the basic anabolic pathways in bacteria. We can see the building of polysaccharides that are used for things like LPS or peptidoglycan. We've got the building of lipids and proteins, along with nucleotides that are needed to replicate DNA. This image looks pretty straightforward, so I always like to add to the end of this unit what true metabolism looks like, which we'll see on the next slide. So here's what all the metabolic pathways in the cell really look like. We've clearly just touched on the surface of the chemical pathways that are used to keep a cell functioning every day. In fact, if you look really carefully, you can see the TCA cycle in the center of the diagram there. If you want to explore this map in more detail, the title of the PowerPoint slide is actually a hyperlink to the website where you can zoom in and out and look at different areas of this metabolism. These are the types of things you get into a little bit more detail in if you take a biochemistry class.